Hello, everyone. Thank you, Armando. Thank you to Park for inviting me to this event. It has been very interesting. All the talks have been really interesting. I'm wondering, do you all see the presentation well?
Yes, we're seeing the
Perfect.
presentation well.
Thank you. So thank you to Gabriela and to Liz, who invited me to this very interesting symposium. A lot of people from a lot of places around the world. My name is Arturo Galván, and I will be talking to you about the effects of different sterols, particularly I will be talking about ergosterol and cholesterol, and their effect on phase segregation. And we're interested in this because of the interaction with polyenes, which I will be discussing in further detail. So as Armando said, we are at the biophysics lab at the Instituto de Ciencias Físicas in Mexico, UNAM. Now, this is the institute. At our lab, we have different techniques. We are always studying the systems with various different techniques. So we have the electrophysiology equipment to measure single channel activity. We have UV specs to particularly to analyze aggregation and dimerization of our particles. We use, of course, the XeBio AFM from Park Systems to do topographic imaging, mechanical properties, and we also have tunable resistive pulse sensing for analyzing particle size and zeta potential distributions. So the outline of my talk is basically just these points. We're just going to give a brief introduction on polyenes and membranes and their interactions. We're going to go into a bit more detail than what Professor Ivan gave in his last talk. I'll be giving also a bit more details on the methods on how we obtain the images, how we analyze the data, and basically the components of the membranes. I'll show the results of the topographic images, the post spectroscopy, the polyene action, and look at a few conclusions. So we study polyenes, but why? Well, in the past, here we go. In the past few decades, fungal infections, particularly internal fungal infections, invasive fungal infections, have become a bit of a problem around the world. Why? Well, basically, we have a problem of a lot of immunocompromised patients, and fungal infections are opportunistic in nature. So when your immune system is low, that's when fungal infections attack. Most of us might get a fungal infection, and 50% of patients with a fungal infection will never know they have it. And that's normally not a problem, but if 50% of people who have it are undiagnosed, and then you have this immunocompromised system, then you have a problem. And antifungal treatments become very important. So normal treatments consist of azoles, which you have here. Azoles basically interfere with the ergosterol synthesis, as Professor Ivan mentioned. One very important difference, two important differences between fungal cells and mammalian cells is fungal cells have a cell wall, more sort of like plants, and they have their main sterol is ergosterol in comparison to cholesterol, which is in mammalian cells. So azoles interfere with the ergosterol synthesis, so that breaks up a little bit of the membrane and also leakage and stuff like that. Flucitosines, like this one, they inhibit the synthesis of essential proteins and fungal DNA. Echinocandins, like this ones, they interfere with cell wall components, and we'll find the last ones, which is the ones we're interested in, which are polyenes. As Professor Ivan mentioned, the main idea is that these molecules make pores in the cell membrane and cause death by leakage of the main components of the inside of the cell. So like I mentioned, the most accepted mode of action is the formation of pores, like Professor Ivan mentioned before. And one of the most important polyenes is Amphotericin B. Amphotericin B, which Professor Ivan already mentioned, it looks like this. It has a hydroxyl chain, a polyene chain, a sugar, and basically the idea is that this polyene is going to go into the membrane, make hydrophilic pores, which you can measure with electrophysiology, like we can see here. There's small little current changes which look like channels, and of course you can see the conductance very well defined here, so that you have the channels. The main hypothesis is that polyenes bind to sterol in the lipid membranes, and these A and B sterol dimers form pores across the membrane, and the interaction, particularly with ergosterol, is more favorable than with cholesterol, and thus the pores are more stable. So you get a better current, a better leakage with the lower concentrations, for example, of nystatin, which is another polyene, very similar to amphotericin B, where in contrast with cholesterol, you need higher concentrations to obtain similar leakage through model membranes, which consist of only just POPC and ergosterol, or POPC and cholesterol. So it's a very simple model where the only difference is the sterol content, and you see a very well-defined difference in the conductance across that membrane. So also, as Professor Ivan mentioned, this is the main hypothesis, but new evidence that has been popping up every now and then in the literature and from our lab too, shows that the sterol is not really necessary, it's not strictly necessary to form the pore. For example, here we have lecithin with no sterol, here we have DMPC with no sterol, and we can see the formation of the channels. We can still see the formation of these pores and the current going through them, even though these membranes have absolutely no sterol present. So this led to the idea that it's not a 1-1 interaction of amphotericin B or nystatin with the sterol molecules, but it's more of the effect that these sterol molecules have on the bilayer, and that's what drives the insertion of amphotericin B and nystatin into the membrane and the formation of pores. And as Professor Ivan already mentioned here, we have a very nice image of where we have more conductance activity, which means more leakage activity in the membrane, and it's where there's a proposed phase diagram that says that this region has a coexistence of liquid disordered and liquid ordered phase. In both sterols, it's the case that this region is the one that has most activity. So this results in suggest that sterols are not essential as 1-1 interactions with amphotericin B, but it's more a membrane structure process that favors the interaction and the activity of polyenes. So this idea of sterol 1-1 interaction with polyenes seems to need some additional criteria. More evidence came from another lab, from Dr. Silva's lab. They are in Portugal, and what they did was they prepared membranes with no sterol, but with phase segregation. Phase segregation means that you have a lipid that at room temperature is liquid phase, and a lipid that at room temperature is gel phase. So these different phases, what you get is different thickness in the membrane, because the gel phase is more ordered, so it's going to be thicker than the liquid phase, which is more disordered. And what they found through leakage experiments of potassium ions is that the more gel phase you have on your bilayer, which is this one is less content, these two are more content, and if you go further down in the fluorescence here, you get more leakage. So what they found was that without sterol, just a POPC DPPC membrane or a POPC sphingomyelin membrane has more leakage when you add nystatin to it, just because you have more gel phase. So this led us to believe that this is also structural, the result of the structure of the membrane, as we thought from our experiments. And another interesting thing is, well, this is gel phase. So If you remember, in the other coexistence phase, you had liquid ordered and liquid disordered. So that gives us an idea that polyenes tend to insert, aggregate, or interact with ordered particles or ordered molecules like lipids, which are in gel phase. And another interesting result that they found was that adding the nystatin to the bilayers, which they formed here on a mica substrate. So these are supported lipid bilayers, which I will go into a bit more detail in how to prepare them. When you add different concentrations of nystatin, the gel phase domains, which are these light areas right here, like light islands, light colored islands, they tend to swell. So this is the gel area and they start to swell. So this suggests that nystatin is inserting into these areas and that's why it's swelling. Same thing happens with sphingomyelin. And in the case of sphingomyelin, there's a surgence, there's a new phase that is originated, which is sort of intermediate. It's not fully ordered, it's not fully disordered, it's something intermediate. So this motivated the experiments, which I'm going to talk about in the next few slides. And the idea here is that, hold on. My computer seems to have gotten stuck a bit. There we go. Sorry about that. So we have the hypothesis that it's phase coexistence in terms of membrane structure that influences polyene activity. So we want to understand how phase coexistence comes about and how these so-called domains, these gel phase areas, which are called domains, have a relation to polyene activity. So in our experiments, I will be using AMB, Amphotericin B, and a derivative which was developed in our group, along with a whole other bunch of researchers from different institutions, both from UNAM and from institutions that are not from UNAM's InvestLab and other universities. They developed this new polyene antimycotic, which in mice has less toxic side effects than Amphotericin B. So those are the ones that I'm going to use. And for the methods, we'll be using atomic force microscopy, which I reckon most of the audience is already familiar with how atomic force microscopy works, so I will not go into any details. I will use the XeBio from Park Systems to use liquid cell imaging of our supported lipid bilayers. We'll have topographic imaging for height measurements, and we also have the force versus distance curve to measure the mechanical properties. I will also use, in some parts, we used the Gwydion software to analyze the data, but most of the analysis was done with XEI, which is Park's analysis software. So what we use? We used POPC, sphingomyelin, egg sphingomyelin. So this is POPC. As you can see, it has one unsaturated chain, whereas sphingomyelin does have one unsaturated chain, but it's so high up in the backbone of the lipid that it's basically a saturated lipid, and that's why it's gel phase when it's at room temperature. We add 5, 20, or 40 mole % sterol of either ergosterol or cholesterol, which you can see they're very similar. There are very small difference between them, but they do have different effects. As mentioned, ergosterol is the main sterol for fungal cells, and cholesterol is the main sterol for mammalian cells. So our model is basically a sort of fungal membrane and a mammalian membrane. So the idea is to see how they interact with the different membranes and how that could be transported to patients or fungal infections. We supported lipid bilayers, which were created via vesicle fusion. So we make liposomes. We basically evaporate the solvent therein. We rehydrate the lipids. We form liposomes via the ultrasonic bath. and we have the unilamellar vesicles. Then those vesicles are deposited on top of a microsubstrate with a total lipid concentration, mass concentration of 0.25 milligrams per milliliter. Depending on the mixture, you have to leave at different times for the lipid bilayers to form. Basically, what happens is the liposome, touch the mica, then they break and they fuse to the mica substrate as bilayers. And that's how you get supported lipid bilayers. We used both contact and non-contact mode. And of course, we used the force versus distance curves. To analyze the force versus distance curves, we also used an in-house program to analyze breakthrough force, which I will get to in a moment. So the data this is the images we get. When we have only POPC sphingomyelin, we see these very big, very clear domains, which we attribute to POPC being on this side, which is lower, at lower height, and this side, which is at a higher height. So this is where we believe this is the sphingomyelin enriched regions. As we add more sterol, you can see that the more sterol we add, the less domains we have. So they tend to disappear as we add sterol to the mixture. And this happens at lower concentrations for cholesterol than for ergosterol, which is quite interesting because this would be sort of like the closest to physiological concentrations of sterol. And in mammalian cells, this would mean that domains are very difficult to see, which correlates to what we find in the literature. The so-called lipid rafts have been very elusive to see right now. So far, they tend to be very small and they are transient. They disappear. They form and then they disappear. However, big microdomains have been observed in yeast. So this sort of correlates really good to what you find in the literature for domains in living cells. When we analyze the height of these images, we do a height histogram. For example, this is the one for sterol-free bilayers. You get a really good peak of the height for the lower part of the membrane, and then the domain here. And as you can see, the more sterol we add, then eventually the domain peak disappears. But interestingly, we found that for ergosterol, the domain peak is higher. So the difference in height, which is we call relative height, between the liquid phase and the gel phase is higher for ergosterol than for cholesterol. When you analyze that in numbers, this is what you get: a very different relative height between the phases for bilayers containing ergosterol than for those with cholesterol. And this is an almost 1 nanometer difference, which is really big when you're talking about bilayers. And also the area coverage of the domains remains quite a bit for ergosterol containing membranes. At 40, there are basically no more domains. To corroborate this, we all used another equipment and another technique, which is frequency modulated AFM, which we did in collaboration with our colleagues from Saitama University, just to corroborate that this height difference was there, it was real. And as you can see from these height profiles, even though the absolute values are a bit different, the difference is the same. It's almost 1 nanometer difference between no sterol and ergosterol. This was basically just to corroborate our findings. From the force spectroscopy, what we did was, when you get a normal, a usual force versus distance curves, you would see this line, and then the red line and the blue line, and they would look pretty much the same. But first, when you have the approach curve and you are on top of the bilayer, eventually you reach breakthrough force, which means the tip is able to break the bilayer and hit the mica substrate. So what we did with our in-house program was analyze the force at which this breakthrough event occurred. And then we made breakthrough or force histograms from that. Since we have the original image, this was done with the help of Professor Armando Antijan from ICF. So since we have the images, topographic images, we can discriminate where we were taking each of the individual curves. So we know if we were on the POPC enriched region or on the sphingomyelin region. So we could color these different histograms to get a better glimpse of what happens. So both sterols affect in a different way the stiffness of the bilayer as the breakthrough force increases for both sterols. Eventually, when you have a homogeneous membrane, you don't have any more domains. There's basically only one peak. So interestingly enough, cholesterol stiffens both the POPC and the sphingomyelin region, whereas ergosterol only stiffens the sphingomyelin region, the already ordered gel phase. Even when you have smaller domains, this is still true. The sphingomyelin region is stiffer in terms of breakthrough force than the POPC enriched region. One interesting thing to note is that even for 40% sterol, for ergosterol, there's still few events that are stiffer than the liquid region, which suggests, and this we confirm, we did confirm this with topographic images, this suggests that there's still small nanodomains that we couldn't see with the resolution we were imaging, but we were able to detect with the breakthrough force. So even at 40 mole percent ergosterol, there are still a few domains left in the bilayer. Doing the grain analysis, which Professor Ortega already mentioned, you get this really nice curve that correlates the size of the domain to the relative height between each phase. So what we can see here is that in order to get really big or really high difference between the POPC region and the sphingomyelin region, you need big domains. But it's always the case that for ergosterol, the relative height is always higher than for the cholesterol containing bilayer. So this is very interesting. So this would be a conserved property, even if you go to very small domains. And finally, the results of the polyene activity. So what I did was I prepared the biolayers, and then I did a solution changeover from PBS to PBS containing one of the polyenes that we wanted to evaluate. In this case, we have the POPC sphingomyelin bilayer, no sterol, and we added Amphotericin B. We can see that the idea was to do the changeover every 0.5 micromolar of A and B to check for any activity, any differences. At 1 micromolar, we could see that Amphotericin B has this sort of like thinning effect. It seems to be like either it's perforating the membrane or it's inserting itself in the membrane and thus sort of thinning, having a thinning effect on the bilayer. And remember, this is at 1 micromolar, and also remember, this is without sterol. So you would have expected for it to have no effect at all if you follow the hypothesis that there needs to be an interaction with sterols. When we use 821 at a slightly higher concentration, at 2 micromolar, twice the amount of A and B, you have the same effect on the liquid phase, so sort of thinning or damaging of the liquid phase, but you also have aggregation on the gel phase. So this suggests that there's a major interaction with the gel phase as well as with the liquid phase, as opposed to Amphotericin B, which only had an effect on the liquid phase. Now we go to the models of fungal cells, mammalian cells, which have cholesterol. If we remember, at 20 mole percentage, there was not much domains to be seen, and this is corroborated by this image here. There are only a very few domains which are the appropriate height, like 1 nanometer. When we add Amphotericin B at 2 micromolar of Amphotericin B in the solution, you start to see this damage on the POPC membrane and aggregation. This is not the size of one domain. This is from 5 to 8. So this is almost a 3 nanometer difference. So it starts to aggregate. So with cholesterol at 2 micromolar, A and B has an effect on the bilayer, sort of like a detergent and aggregation effect. In the mammalian cell model, A21 does not make any sort of holes, but it starts to aggregate very much. And as you can see, the original image had defects on the membrane. So these are holes that go straight into the mica surface. I usually try to leave these features so that I can really evaluate that our membrane is a bilayer and it's not a multilayer. So these holes disappear when you start adding A21. So this suggests either two things: either the holes are getting filled with A21, or A21 somehow is inserting itself in the bilayer and thus swelling it, and the holes disappear. Unfortunately, with just topographic images, we cannot decide which of the two it is, and we can only evaluate aggregation. And this happened at a higher concentration than for Amphotericin B, and even at a higher concentration than A21 with POPC, sphingomyelin, and no sterol, 5 micromolar of A21, which is pretty high. Finally, for the fungal model, we have the domains at 20 mol percentage ergosterol. We remember we still have some pretty nice circular domains. Amphotericin B has an effect at 1 micromolar, which is just aggregation. It doesn't seem to have any effect on the liquid phase, but on the gel phase it aggregates. So it seems to be inserting itself in the gel phases and starting to aggregate there, but still there's no damage at this concentration. In the case of A21, we have two different phenomena. In this case, I did add another concentration because of the following phenomena. First, as you can see, when I add 1 micromolar of A21, the derivative, which is less toxic, the gel phase domain swells. So this happens also with the results that I presented to you from Silva and her group. So this suggests that A21 is actually getting inside of the gel phase domains, and it's swelling them. Okay, so I wanted to see if this swelling effect carried on with more concentration of A21. So I did another concentration, and you can see that there's a slightly difference in the swelling effect of the gel phase. But also at this concentration, there's really big damage done on the POPC liquid phase. So this suggests that A21 also has a detergent-like effect on the POPC phase, which is liquid. So to summarize what I've been presenting to you, so ergosterol and cholesterol modify the dynamics of domain formation and bilayer thickness in slightly different ways. Ergosterol increases bilayer width, particularly of the ordered phase, which is the sphingomyelin enriched region. Because of this, we believe that ergosterol seems to interact favorably with sphingomyelin, whereas cholesterol seems to interact with both POPC and sphingomyelin. But this is from the breakthrough histograms. Relative height between phases depends on ordered phase area, the size of the domain, and is always higher for ergosterol containing membranes. That is really interesting. It doesn't matter if the domains go very small. If there's ergosterol, the relative height is always higher for ergosterol containing membranes. And finally, A and B and A21 present slightly different activities on the bilayers that we studied. A21 has a bit more of a detergent-like effect on ergosterol as compared to Amphotericin B. And in cholesterol-containing membranes, A and B has a detergent-like effect, whereas A21 does not. Both polyenes present an effect on sterol-free bilayers, which is interesting because there's no sterol, and A and B has a thinning effect, whereas A21 has both detergent-like and aggregation effect. So this last one is also in accordance with this last studies that I have presented to you, that the sterol is not strictly necessary for polyenes to have an effect on the membrane. I would like to thank all of the people involved in all of this work, people from UNAM, particularly Professor Ivan Ortega, who is the leader of our group at the Biophysics Lab, our collaborators at Setama University, Fernando Favela from Tecnológico Nacional. He does most of the simulations that we work with, people from the University of Groningen, senior staff, and finally from UAM. Most of these worked with the development of the A21 derivative, which is less toxic. I would also like to thank our funding agencies, CONACYT and the GAPA, primarily our main sources of funding. And with that, I would like to thank all of you for your attention.